Okay, well, thank you very much for inviting me this evening. I'm head of the Computer Graphics and Visualization group at University of Bedfordshire, and I've been there for 7 years now. And we've evolved, so we've got probably about a dozen postdocs working on various things. We're currently engaged in 9 European projects, and those are mostly in the area of medical visualization. So that's the focus of the talk tonight. What I'd like to do is, first of all, talk about the background, which is the Virtual Physiological Human, and I'll explain what that is in a minute, and then look at the activities that we're doing in general, and then focus on 1 or 2 applications which we've specifically done within the projects in the last few years to give you an idea of the type of thing we get involved in. And please interrupt and ask questions at any time. Okay, so the physiome. We're into biology immediately. Okay, physiomes are quantitative and integrated description of the functional behavior of the physiological state of an individual or species. In other words, it encapsulates the entirety of what's going on in any species at any time. Okay, so it's a model of what's happening within that species in its entirety, at all physical and at all temporal scales. So it describes the dynamics of the organism, what's going on, and why. Comes from life and as a whole. So it reflects the entirety of the body. So it defines relationships right through the organism, going through multiple spatial scales, and the various interrelationships amongst the constituent parts. So when we're trying to describe this, we're looking at integrated models, and generally when you're talking about healthcare, for example, you tend to think of things in terms of organs. So you go to the cardiac department, or you go to the renal department, or something like that, depending upon what the problem is. We're trying to get away from that and integrate people who previously didn't talk together, because a lot of interrelationships within what's going on in the body, and you can't just cut things away and treat them in isolation. Problem is that that's complex. Okay, so life is multi-scale. You will see things happening at many, many scales, going right down through the genes, the molecules, etc., and working your way back up to the whole body. We tend to think at an organ level when we're talking about biomedicine, but there are lots of things going on at different levels. So it's complex, and they're taking into place at different spatial scales and at different temporal scales, going from a lifetime right down to the molecular interactions that are taking place within your cells. So we can try simplification and introduce a simplified human model. And it may work. You know, you can get some success with a simplified model. As you can see, we do get some successes from a simplified model. However, in general, the human puzzle is a bit of a mystery. We need to put the bits together in an appropriate way. So we're investigating at the moment this jigsaw puzzle made of 1 trillion pieces by picking a little bit out and shaking it around and looking at it and trying to put it back in place again. You know, and really that's not the way that things are going to work well, I think, into the future. So we're trying to create something, a framework within which we can put these constituent parts together. It's not going to produce the whole picture, but it's going to reflect more the whole picture than we currently have. So we're going from complete reductionism, where you cast things into their constituent parts and they're all working independently, churning away inside there, to integrationism, where we're trying to we now put the picture together. We hope it makes sense. So integrationism and they fit together. You can't do one without the other. So integration, what we mean, integration across subsystems. So different parts, the endocrine system or the digestive system, cardiovascular system, etc. Across scales in terms of dimension, from the organism right through down to the tissue, cell, the atom, and in temporal scales, because things are happening at different time scales within the body, and across disciplines. And this is quite important because often there are barriers People in different disciplines work in different ways. They have different conventions. They use different units. The way of thinking of data is different. And getting this communication going is a work of art in itself. Okay, so what the Virtual Physiological Human is trying to do, it's an integrative research approach, trying to create a framework of technology and methods. We're not trying to produce a model of the human physiology and you press the button and out comes the answer as to what's happening in that person. What we're trying to do is to establish a framework and build up a picture. So all the various constituent parts will be able to talk to each other. You know, there will be a common framework in which you can use data from different researchers and ensure that it's compatible with what you want. And this is a long-term thing. So, you know, we're looking 20 years. It's the type of timescale Commission is thinking of in supporting this framework. So what we're trying to do is to make it descriptive. In other words, it says what's there. Integrative. In other words, it pulls things together. And predictive. So if you have some data and information, you can draw some conclusions from that and to identify what actually the outcomes are likely to be. And obviously for clinical application, where this is headed, it's the predictive is the critical aspect. Okay, so we have the DIKW pyramid, where you start with data, you build that into information, you put it into knowledge, and eventually, if you're really lucky and you live long enough, you end up with wisdom. Okay? And this is what we're trying to do. We're taking the data, we're trying to provide additional information on top of that. We've got metadata, trying to support what's there, and then from that we're trying to put in some predictions, some modeling, from which we can gain validation, confirmation, outcomes from the type of structures we're building. We worked a lot in musculoskeletal modeling with orthopedic surgeons and things in Italy. And the concept is here in that, where you start with the organ, which in this case is the limb. You go to the constituent parts, the bone. Within the bone, you've got different types of cells. So you've got the sheath cells are much harder bone. This is spongy bone, which provides some sort of impact resistance at the end of your bones. And then you work down within these. You've got trabeculae, which are the holes in the spongy bone, and you can go down into the cells, etc., etc. So all of these, you have people working at these different scales. We need to integrate the information that's coming out of those. So the implications of what's happening are, firstly, we've got the data explosion. I mean, talk to any computational biologist, anybody working in biomedicine, they're all complaining that they can't cope with the data that they're getting. Data capture devices like scanners, MRI devices, CT scanners, etc., are increasing in resolution, so the files are becoming bigger, they're providing more information, and the data sets are becoming larger. You're getting many more modalities coming in, so for an individual patient, you get a multiplication of the types of data sets that are available. And if you're working in a multi-scale problem, for each individual problem, you've got now multiple sets of data, each at different scales. So the overall data demands are escalating. You've then got a problem of data compatibility, because you're capturing data from different devices, possibly at different locations and different laboratories all around the world, maybe using some old data, some newly captured data. Trying to ensure that you've got data compatibility is becoming a bit of a nightmare. And you've got registration issues where you're fusing this data and trying to create a general picture out of it. So data is often captured at different sites, and the computational resources which do it are often different. You just look at the operating system, for example, and how using the software, etc. You will get obvious incompatibilities which you have to be overcome. And when you're doing simulations, you have to ensure the boundary conditions are compatible across the multiple simulations that you're performing. The computers and infrastructure supporting this now tend to be distributed. So you have data in databases scattered around internationally. Software is often located with the data or on distant servers, or you may be using grid technology, this type of thing, where you're using multiple servers to do things. So distributed resources are becoming very common. So you've then got to have an architecture to support user access. So that's good allocation of time on high-performance computers, for example. You've got to book ahead. You've got to ensure that that's going to be available when you want it, which isn't necessarily what the clinician wants, because the clinician may have an emergency that they really need the data here and now, and getting access to some of the high-performance computing resources is not necessarily very straightforward. The more data you have, the more difficult it is to find what you're looking for. So you need a semantic search, so you need annotations, you need ontologies and that type of thing to support your search. And when you found the data, how do you know what it is? Whether it's the data that you want, well, you probably need some previews associated with that to enable you to determine that the data you've actually discovered is the data that you really require in your circumstances. And interoperable, flexibly combined software. The idea would be that you try and produce some sort of workflow, so produce a complex task, and you build that out of components. If you're going to do that, then the components have to be integrable. They have to be compatible. What's being passed on from one of the components to the next has to be appropriate. And different scales may require different degrees of computational power. People working at the molecular level are often running high-performance computers for days on end, produce single simulations. And if you're in a clinical situation, particularly, you've got problems with security, firewalls, hospitals don't like their data being exposed to the outside world, etc. So you've got to overcome all of that within the work regime that you're trying to create. So the impact on healthcare. What the Commission is very keen on is patient-specific medicine, and that's generally a worldwide trend. You're trying to get away from the generality of clinical approach to focusing on the individual and what the needs of the particular individual are. So the availability of multi-scale data, you then have to combine it in a particular way and make it available to the clinician in a way that the clinician is able to interpret. The data may be collected only by highly invasive techniques. In other words, you know, you get killed by the data collection, if not by the disease you're trying to resolve. So often the data will be collected only from cadaverous tissue, and that will be made available then by some generic model which you can bring back using an atlas or something similar. So this is a way of building up a larger picture, getting data that you wouldn't normally be able to get from a patient, but being able to adjust that generic data and fit it to the individual patient to get a closer match to his circumstances. Genetic data may be relevant in some conditions, that has to be factored in. And sometimes population data, where you actually get sort of what happens to a male age 45 with his particular profile, rather than this particular individual, is integrated into the solution. So what would be nice for the clinician to have is to be able to set up a what-if scenario. At the moment, it's very much suck and see, particularly if you're applying multiple drugs, for example. It's very, very difficult for the clinician to predict the outcome of a number of drugs being applied simultaneously on any individual. So it would be nice to have a model that the clinician can actually try what's going to happen if I combine these treatments, what's the likely outcomes. So that prevents a possible disaster into the future. Okay, so that's the general background to where it's going. So the VPH is a program within Framework 7. It's likely to continue into Framework 8. The Commission has already put 200 million euros into it in Framework 7. And as I say, it's likely to double that in the coming future. So they see it as a key ongoing priority within the framework programs. We were very much involved at the start of VPH, and we co-edited the roadmap which defined for the Commission what approaches they should take and how they should organize the ongoing VPH research and what priorities it should take into Framework 7. Okay, so now I'll look at my research group and see the type of things we're doing in general before we focus on specific applications that we do. So I look after the mainstream graphics, visualization, general modeling, computer systems, etc. We also work a lot on GPUs, graphics processing units, and GPGPU, which is general purpose programming use of GPUs. Come back to them in a minute. Web services, because when you're dealing with an integrated environment, you have to access data, information, etc. At a distance, and web services are a standard way of bringing this to you and integrating it into workflow. Image-based approaches, where you're taking imaging, manipulating images, and using that in other aspects of computer graphics to generate images. And we've just started a computer games research area with Professor Prakash, who's joined us within the last couple of months. In fact, VPH spans most of these, even games, because games technology is now being used even in the medical area. There are lots of very good fast modeling techniques for surfaces, use of things like the Wii controller is a very, very intuitive device that can have its applications. And games engines are very good at producing output very rapidly for trying things out to see whether they work or not. Our current projects span a number of areas. What we spent in the last sort of 7 or 8 years is some time developing an underlying framework of software which will support a variety of applications and making that robust. We worked with international partners, and now it's used in a number of areas around the world. That's enabled us to move into applications. So we now have a framework which will support a variety of applications. It's very, very versatile and was designed to be so. So we look at projects on osteoporosis, heart failure, brain and lung cancer, also operational activities such as placement of stents, as well as some sort of more fundamental use of GPUs and some community actions in which we're trying again to promote discussion and development and identifying where the trends ought to be and how we should be developing into the future to support where we see the research going. The general description of the work is we supply tools. We have a user base. We try and assess what their needs are in very close consultation with them and supply tools which suit the requirements that they have at any particular time. So we work very closely with them. And the tools may mostly involve visualization because that's our strength, but lots of modeling or image analysis, that type of thing. Also supply web services, and we can re-engineer existing software to take advantage of GPU technology or to use games technology to increase speed where it's relevant to the problem. So all our activities are essentially focusing on users, and if users come to us, then we try to assess the problem and apply what skills we have to finding suitable answers. In terms of medical visualization, the data normally comes from medical imaging. You've seen the things on TV, I'm sure. Different modalities are good for different things. So CT, for example, is good for bone because the bone stands out from the background, whereas it doesn't distinguish very well amongst the various types of soft tissue that are there. If you want soft tissue, then you would tend to use an MRI. Very familiar to most of you, I would imagine. In this, the data is provided in 2D slices. So you see a slice through the body, and you get a stack of these slices which build up into a 3D body. So it's like a stack of playing cards, if you like. You get the individual images and you put them together and you create a 3D structure, which you can then interrogate as a 3D structure, not as 2D images. So the readings of each of the slices are interpreted as grey values. So you can see this grayscale image. And each tissue type has a characteristic grey value which enables us to distinguish between the tissue types. There are mostly two forms of visualization. One is a surface visualization where you look at the outer structure of an object. So if you had the heart, for example, you see the outer surface of the heart. So that is, the surface is where the heart tissue changes into not heart tissue, if you see what I mean. Okay, so it's the transition point between two types of tissue. So what you're trying to do is to identify a boundary between two sets of grey values. And the trick is to get that boundary value right. And if you can identify where that lies, then you can sketch a contour of where it is in the slice and build those up into a 3D surface. The alternative is volume visualization, where you take the volume as an entity and look at the grey scales, and you associate with each of these grey values certain visual characteristics. So, for example, this grey value tells me that this is heart tissue, and I want the heart to be red. So every time I find that grey, I will put in a corresponding red item at that position. Okay, and there are various other optical properties that you can associate with it. So transparency, do I want to be able to see through it? If I completely turn it totally transparent, I will take it out of view completely. If I make it opaque, then it hides everything behind. So it's your choice as to what the visibility is going to be, and various other surface characteristics that you can build into to provide a particular graphical effect that you want. So those are the two types of approach you would have for rendering views from this data. What you find is visualization is normally computationally demanding. So methods will often use shortcuts in order to be able to get through to the output. What you find is that changing the viewpoint is fairly straightforward. Zooming around in 3D and looking at it from different perspectives is straightforward because the underlying structures are exactly the same, not changing anything. So that's a fairly straightforward thing to do. But if you're changing the isosurface value or the transfer function, then you're actually fundamentally changing what you're trying to display. So the underlying model is changing, and that's then more computationally demanding. And that's when people try and perform some tricks in order to speed things up. GPU technology has taken a lot of the sting out of the computational demands, but what happens is that you have to know what you're doing. You have to understand the GPU architecture because it's different from the CPU. So you have to think your way through what you're trying to do and map it very carefully onto the way in which the GPU is constructed. If you don't do it, then you lose an awful lot of efficiency. So in general, what we're trying to do when we produce the image is to use ray casting as our standard approach. And if you're using ray casting, you're throwing rays into the medium, and those rays will impact on the objects in the scene, and that will determine what you're going to display at a particular point in the image. Okay? So if you can arrange things in this parallel processing GPU environment so that all of the rays have an equal length, then they all in parallel process over the same time, and your stepping forward takes place in synchronization. If some of them have long segments, then the short segmented rays have to wait until the processing on the long segments has been concluded. So the more uniform that you can make your rays, the faster your processing takes place. Ignoring empty voxels. So if there isn't any material there that you want, don't process it. Use proxy geometry. We have replaced voxels by spheres, and that can produce a considerable simplification. Use view-dependent sorting for occlusion. So if things are not going to be visible, then sort them out. So if you know that they're behind something and that is totally opaque, don't process it. You have no need to. More accurate ray cell intersection for improved quality. Mostly you will find the methods, when they're looking for intersections, will use linear interpolation, which is crude. It produces artifacts. You'll see some of the artifacts that it produces a little later. What we try and do is to use a higher degree of interpolation, and that produces a much smoother result. The problem is that's costly, so we've found ways of getting around that computational cost and overcoming the bottlenecks associated with it. We've got multiple transparent isosurfaces. This is new. So if you've got an isosurface, in general, GPUs will only give you one transparency. Otherwise, it costs you in computational time. Our approach gives you 256 levels of transparency at no additional cost, simply by the way in which we arrange the computation. And a lot of that is use of empty space skipping. So if you know something isn't there, you don't keep investigating it. You go to the next stage that you want to look at. So that's general background. Now, if you can see the quality comparisons, this is probably the one that stands out most easily. This is a backpack, MRI of a backpack. This is a can down in here. And a standard approach using finite differences to get the intersections, you get a lot of artifacts on this surface. The standard approach using a Sobel filter, which is a higher resolution of the you still get these artifacts, but on ours, most of the artifacts have gone away because that uses a higher level approach. It doesn't use the linear type of intersection that the other methods do. So I'll look now at the application areas that we're involved in. First one is VPH2, which is VPH2 is actually the virtual pathological heart or the virtual physiological human. That's it. So I keep forgetting it. So that's why it's VPH2. It looks at the left ventricle. This is the anatomy of the heart, built in 4 chambers, the atrium and the ventricles. The left ventricle is the one that does the work. That's what pumps the oxygenated blood around your body. If you go into left ventricular failure, you've got a problem. So what happens normally to cause left ventricular dysfunction is that there's been a heart attack, some of the heart wall has been starved of oxygen, and the cells have died. So there's dead tissue within the heart, and it's now no longer functioning in the way that it ought. So when that happens, it tends to enlarge. So it starts filling the cavity in the chest. It becomes more spherical. It's therefore less productive. Even though it's got less tissue, it's even less productive than it would otherwise be. And the problem is that when this ventricle is pumping the blood out through the aorta, these valves should close to prevent regurgitation back into the atrium. Yeah? If this becomes more spherical, then this pulls the valves apart, and it allows blood to go back into the atrium as well. So there are a lot of things going on at the same time which are causing problems once you get the initial heart attack. So when people get into heart failure, the cardiologist and the cardiac surgeon have to make some very, very critical decisions, and currently there are no tools at all available to support them. They use their judgment, essentially, and their experience. So what we're trying to do is to develop, in association with cardiac surgeons in Italy and modelers in various other parts of Europe, is to create a model of the left ventricle which will enable surgeons to make a decision as to where they should cut and what other restorative actions they should take. So we're creating a model of the left ventricle. Unfortunately, the data is of very low resolution. The slices that I talked about are 8 millimeters apart, which is a lot. So you can see that the data is separated by quite a lot, and therefore even get starting with a suitable model has taken us quite a lot of time to develop methods which provide a surface which is suitable for further work. So once you take the MRI scan, and remember when they're taking the scan, the heart is beating, it's not stationary. So they're taking a slice and they're trying to take the next slice at a similar stage in the cardiac cycle and work your way down the heart. Meanwhile, the heart is jumping around, up and down as well. It's not stationary. So actually sort of capturing this data is a bit tricky. And then they inject gadolinium, which is a poison, but it works as a stain to identify where the necrotic or the dead tissue is. So you then got another set of MRI, which you can map onto the first set of MRI to identify which areas of the heart have died. Okay, so you've got 2 sets of data that you have to register them. The problem is that they're not always taken at exactly the same time in the cycle, so registering them isn't a straightforward process either, but you end up with an area which is seen to be of interest to the cardiac surgeons. Okay, so now they have to decide what precisely they're going to do. So we give them a tool which enables them to define the type of incision that they're going to make in order to remove the necrotic tissue. And in consultation with them, we give them various fundamental shapes. So they can adjust this, and when they're planning, to pull at any of these to put it into the position that they want, and then decide that's the cut they're going to have, and that part of the wall is removed. So that gives them a feeling for what the outcome is going to be as a result of their incisions. Okay, they then have another choice. Do they just close the heart back up? Problem is, if they've removed a lot of tissue, the ventricle is now much smaller than it previously was. Can it actually cope with pumping the blood around the body sufficiently well? If it can't, if it's likely that is going to be too small, then they can put a patch made of synthetic material, which is stitched on the inner surface of the heart, and then instead of stitching the two parts of the heart together, the outer sides of the cut are stitched to the patch. Okay, so again, this system will apply the stitches automatically and apply a force model for them to see the ultimate shape of the heart. What they're trying to do is to bring the heart from the spherical shape that it was in failure back to a more ellipsoidal shape, which is more comfortable for the heart chamber and more economical for pumping. Okay? So that's where we've reached at the moment. The next stage is to register the shape with the heart vasculature. In other words, to know where the blood vessels in the heart wall are, and to be able to map that onto the model as well, because sometimes the surgeon will do what they call revascularization. In other words, they will enable the blood to flow through the veins more efficiently than it currently does, through the heart arteries, not the veins, and to enable the heart to function rather better. Okay, so they will often do that at the same time as they're performing their major surgery. That's what we still have to do. And the second thing we have to do is to predict the post-operative situation. So having performed that operation, having decided that's what you want to do, okay, so what is now the volume of the heart? How efficient will it be? How will it cope with the type of forces that it's trying to produce during its subsequent activity? Okay, so that project will run until towards the end of next year. We've got that time to conclude that and then perform tests and let the clinicians work through and decide any changes that they want. Okay, another project is called Contra Cancrum, against cancer, if you're a Latin scholar. And this is to develop an advanced multi-scale problem looking at tumor growth and response to treatment. So you have a tumor, and how will it react to radiotherapy? How will it react to chemotherapy? So CCGV tasks have involved modeling, visualization, web services, and GPUs. So we've had the kitchen sink thrown at this project on the various aspects of the work that we do. We look here at the GPU work to give you the type of idea of what GPUs can do in an application situation. Okay, so the simulation method that's applied is essentially to look at the volume surrounding the tumor and break it down into zones. What they call them is geometric cells. I'm sorry that they use the word cells because that's very confusing, but I tend to think of them as zones. So they're cuboidal zones surrounding the tumor. Okay, and they have a starting situation where they know what the types of cell are in each of those zones. So each zone will contain a number of biological cells of differing types. Healthy, cancerous, on the way to becoming cancerous, etc. Various different categorizations that they will have. And then there is what they call a cytokinetic diagram, which describes how those cells will evolve. So they go through various stages depending upon in what form they start. Okay, well obviously in a tumorous situation the cells are multiplying, so when that happens the cell becomes, the zone if you like, becomes overloaded with cells and they burst out into neighboring zones. And that again is modeled as part of the process. So it's a time-stepping algorithm where you look at the current situation, you perform calculations as to what is going to evolve to in the next time step, and then adjust the number of cells in each zone by shipping them to neighbouring zones if the excess number has been exceeded. Okay? And you run that through a number of time steps, 500 or 1,000 time steps, and you can see the evolution of the tumour. So this is a technique that's been used for quite a number of years by the partners in our project, and it's been quite successful. The model is fairly accurate. However, it seemed to us that computationally it really wasn't very efficient. Just look at the GPU. The GPU is started off as the thing that controlled the display in your computer, and as computer games became more and more predominant, it took over a lot of the processing of the visual elements of your computer, and now it's by far the most powerful processor in a standard computer. If you look at the evolution of the GPU compared to the CPU over the last 10 years, then in terms of gigaflops, in other words, how many floating point operations you have per second, performance, you see that the blue lines are CPU development and the green lines are GPU development. Okay, and if you look at the memory bandwidth, in other words, how efficient it is at moving information around, again, you've got the CPU and the GPU are diverging radically in performance, and it's the trend is accelerating. So the GPU now represents an incredible powerful resource on your desktop, but using it for general purpose programming isn't always easy because of you have to recast the algorithms to take advantage of the GPU architecture. So simulation that our partners were using previously took 20 minutes on a desktop computer. Okay? It's a Monte Carlo approach. So essentially what they're looking at is the development of these cells, but the movement of the cells from one zone to another is randomized with a statistical pattern. They know what's likely to happen, but you can't predict exactly what's going to happen in a disease like cancer. So what they do is to run it thousands of times and then produce a statistical overview of the likelihood of the evolution of the tumor. So that means that that's one run, but they've actually got to run it lots and lots of times. So if they can save time, then it's going to make a remarkable change in what they can do. So what we found is the more complex the problem, in other words, the higher the resolution, the more zones you have, or the more time steps you employ, the more you benefit from using the GPU. It isn't a linear change, it's better than linear. So the use of the GPU reduced the time to 8 seconds from 20 minutes. Okay, that's just that's the first shot we had. It's a speed-up factor of 150. We also got a Tesla S1050, which is an off-the-shelf GPU cluster. It's 4 GPUs in a box. You plug it into the back of a standard desktop computer. No technical knowledge required. Using one of those, reduce the time to 2 seconds. Okay, which is a speed-up factor of 500. This is old technology. This is a 2009 GPU. The 2010s should be able to double that factor quite easily. Okay, so that's the type of benefit you can have because the problem mapped very well onto the GPU architecture. Why? Because it was cell-based, parallelizable, and therefore we could map it very well onto the features that the GPU exhibited. Okay? But what that means is that they can now deal with their data in a completely different way. Instead of going off and making 2 cups of coffee, having a sandwich and asking their friends around for lunch, they can just press the button, out comes the answer, look at what happened, are the parameters right, is that really what we want to do, and have another run almost immediately. Okay, another example. We've worked a lot on muscle wrapping, the musculoskeletal system. So this is the osteoporosis project, which I'll describe a little in a moment. But when biomechanics people model muscles, traditionally they've used action lines. Muscles are very complicated, and they simplify them to just simple linear descriptions. Mostly these action lines are straight, joining the 2 points on the bone where the muscle is inserted. Okay? Sometimes muscles aren't nicely, conveniently straight, though. They wrap. They wrap around other muscles. They wrap around bones. So in that circumstance, the linear line of action is no good. You have to look at something a bit more complicated. So we've developed an approach which enables you to find the shortest path automatically around the obstacles of the type that you will find in bone. So we model them as spheres, cylinders, multiple cylinders, etc. And that seems to do a reasonable job. So that's mapped the action line, which enables you to see where the muscle is going to go during the motion. But that's just the starting point. So starting from that now gives us a shape on which we can build the formal shape, which is the muscle. Okay, so as the limb moves, so the muscle changes in shape. And that shape is very much associated with how the action line moves. So we're now developing a pattern. This is very, very early stages of the work. The project has really only started this month. This is our sort of preliminary investigations, and it's going to take another 2 years to complete this project. But that will enable us to work out what's happening to the muscle during its motion. This is new. This isn't available anywhere else at the moment. You know, this is groundbreaking stuff. But beyond that, we're going to one step further, and we're looking at what happens to the fibers within the muscle. Because this is important because muscles are not isotropic. Yeah? If you pull along the muscle, that's where the strength is. That's where the run of the fibers. Across the muscle, there is no strength. They're not applying any forces. So it's important to know the directions of the fibers of the muscles. Why do we need it in this project? This project is on osteoporosis, which is brittle bone disease is one of the common names for it. It's particularly associated with older women. The bones tend to get fragile and the skeleton gets very damaged in the vertebrae and on the hip, often associated with hip fracture. We've got 20 partners around Europe looking at various aspects of osteoporosis. It's the first large-scale multinational project looking at osteoporosis in its entirety. So previous treatment for osteoporosis has been fix the fracture when it happens. However, the fracture occurs in elderly people in general. It's a life-changing occurrence. It changes their mobility. It may mean that an independent person now has to go into care. Osteoporosis actually kills more women in Western Europe than breast cancer every year. The reason is that they fall and the shock is sufficient that their system can't cope, and a few weeks after the fall they die from other circumstances. But essentially the trigger was the fall associated with the break of the hip, for example. So, for example, we've got partners in Charity Hospital in Berlin, and with the agreement of patients, they have put a hip prosthesis in when they've done a hip replacement, which is electronically monitored. So it has 4 sensors within the prosthesis, which enables them to monitor what forces are taking place. So they get these people back regularly in the laboratory and just ask them to perform general movement so they can see what forces are taking place within the body. Very difficult to achieve apart from in a cadaver otherwise. And what they've found is just occasionally they get a stumble. An innocuous stumble, something a bit like that. And they have been absolutely staggered by the size of force that this generates within the skeleton. They've had registered 8 Gs, for example. Phenomenal amount of force for such a small occurrence. So now you can see if the person has a vulnerable skeleton, this is going to cause a fracture. So they have films of several of these occurrences, and all of them are remarkable. They're different, but each of them is eyebrow raising. So that's where we're going. So the emphasis is moving from treatment after it's happened to prevention. So one of the other partners has developed a belt that a patient can wear for a week. The belt has got gyroscopes in and is able to monitor the type of activity that person is undertaking throughout the week. So they've got a recording device within the belt buckle, and that enables them to capture what's happened to that person during the week. And therefore you can see whether there's anything in the person's lifestyle which is likely to trigger a problem, and therefore counsel them to avoid that type of action, etc. So it's a wonderful project to be involved in, because all of the partners are contributing remarkable technology to it. Some of the other things we looked at are large volume data. This is beginning to be associated with our GPU work, but say large data sets are becoming much larger than they previously were. And in some circumstances, the data sets are larger than the available memory. So if you've got an XP operating system, for example, your data set is 4 gigabytes tops. Go above that, you can't cope. Okay? So if you're trying to use a data set which is larger than that interactively, then you've got a problem, because you can't actually put your data into the computer and use it. So what you have to do is to ship the data back and forth from memory to your hard disk as you're interacting with it, and try to do it seamlessly so that the user doesn't notice. Yeah, okay. So you have to be very careful in the way in which you structure your data. And the approach that we used was the fact that if you're looking at a body, and you want to see the whole body, then it's unlikely that you want to see the detail. So the body is represented in less detail than is available in the data set, because you don't notice the difference. But if you want to zoom in and see the detail, then you can't see all of the body. So you throw away the bits that you can't see and store the detail of the bits that you can. And I'll show you a video of that happening. And just on a standard desktop computer, we've been having interactive speeds with 24 gigabyte data sets without any problem, by using a structured approach to the data. In other words, we get the data, we restructure it because we know its character, and then we're plucking it in and out of hard disk in a predictive way, so we know exactly how best to put the next set of data and make it available to the user and be ready when they want it. That's called out-of-core techniques if you're not familiar with them.
With the solid-state hard drives that are coming along that offer much higher performance than regular hard disks, do you see an application for that sort of technology in this?
Yeah, because it's not so much the hard drives, it's the fact that the operating system can't address more than a certain amount of the data. In 64-bit system, that's become less of a problem. On GPU, it's still a major problem. So what we did is we developed the technique on the CPUs, and it works, but now we're shipping it back into the GPU, so we're going to be able to use large data sets on GPUs, which normally have very restricted memory, 1 gigabyte or 2 gigabytes or something like that. But it isn't the capacity of the hard disk or the speed of the hard disk, although obviously if you've got a faster hard disk it works better. It's the actual bound of the memory available. So in conclusion, and I'll show you a set of videos after this if you have time and you're all still awake, is the virtual human here? No, clearly not. But there's a huge effort going on, and keep your eyes open, because there will be significant developments in biomedicine, and one would hope that you'll begin to see it percolating, not maybe into the GP surgery, but it may start to influence clinical medicine in the more advanced hospitals within 5 to 10 years, one would hope. More general application, probably you're looking at 15 or 20. Doctors are very conservative, and rightly so. They need to know that it's working before they'll change their previous practices, and you can understand that. So they want to see some proof before they'll get behind anything. Okay, so changing of thinking. What we're finding is that it took researchers a few years to grasp the idea and to throw away their preconceptions and realize that they had to work in a different way and collaborate across these scales. It's now happening. The Commission has put a lot of momentum into it, and for once the Commission has done some good work, I think. It really has helped to drive the technology. And it isn't just a European effort. This is going international. There's a large effort also in the States, and we've got a number of projects with partners stateside like Stanford University also developing this and pushing this further. Okay, so I'll maybe take 1 or 2 questions now if you want, and then I'll show some videos when those have run out.
In developing your visualization approaches, what you're doing has a lot in common with the seismics industry, for example.
Okay.
Do you collaborate?
No. Yes, well, precisely. It's transdisciplinary. I don't know anybody in the seismic industry. If there is common ground, I'd be very interested in exploring it. Well, we've been working with an oil company on developing oil exploration software recently, but that was different from seismics. Yeah.
I'm interested in the way they correlate the heart image slices, considering the dynamics of the system. I mean, not deeply mathematically, but what's the generic approach for making sure that the actual slices coincide between the before and after version?
They slices in before and after version. Sorry,
the before and after the injection of the dye, because you pointed out that the slices were taken at different points on the cardiac cycle.
The two are captured at different times.
Yes. So
Okay.
how is the correlation performed? The equivalent slice, so to speak, before and after dye injection, because they'll be on different parts on the cardiac cycle inevitably.
They attempt to capture each slice at the same point in the cardiac cycle. Okay, so they're not all captured simultaneously, but they are captured at equivalent points in the cardiac cycle in both technologies. Okay, so it doesn't perfect, because we do see, differences in the data that you can clearly see, and we have to try and work around that. And then, having got those two models, you have to map the one onto the other and register one surface against the other, which isn't straightforward, because there are very, very few landmarks associated with a ventricle. So mostly the landmarks you have to work with are where it joins to the right ventricle.
So my question essentially is about that mapping.
Yes.
I mean, what's the generic, sort of broadly speaking, the mathematical process you use for doing that mapping?
Well, it's just essentially what you have a registration, and you're working with two clouds of points. Yeah. So you're trying to register one cloud of points against another cloud of points.
So it is in fact it's a spatial mapping, which the GPU is of course ideal for.
We don't use the GPU for that.
You don't use the
we
GPU?
do that on the CPU. Yeah.
I was thinking basically of war games, because that's exactly done, isn't it?
Yeah. There isn't need for huge speed of that process. Accuracy is important, and it's done offline. So you have you would probably have a well-trained technician performing this process, ensuring that it was happening appropriately, and they would take time and care to do it well. So you're not rushing to throw data at them.
How far are you getting, or have you got, in terms of the interaction of various organs and trying to cross-refer them? For example, if one looks at the way in which foodstuffs move through the body from the stomach, and then you go into the liver and the kidneys and the pancreas and so on, there's a lot of interaction going on there. How far are we down the line of
very little down the line. Essentially, most of the projects are still based on the old structures. So you have a heart project, you have a renal project, etc., etc. But what they're trying to do is to work within each of those confines at a multi-scale level. In other words, you're pushing this out, if you like, into a three-dimensional matrix of organ, scale, time, etc. And we're working along certain channels at the moment, and we're not moving in other directions in anything like the same speed. So mostly it's physical scale at the moment, and getting people talking to each other who previously hadn't, particularly molecular scientists with the clinicians.
I think your 10 to 15 years might be optimistic.
Well, it depends. You're not trying to get the answer, the answer. What you're trying to do is to set up a structure which becomes continually richer. So you populate it with more and more sophisticated models, and populate parts which have no models at all at the moment, or link models that exist at the moment. But it's going to be patchy, and it's going to evolve in fits and starts. But it doesn't mean to say that it's totally useless until the whole thing's there. Parts of it will be usable. And so they're mostly concentrating on heart disease and cancer at the moment. Obviously, in Western the societies that tend to be the killers. And in those types of projects, with enlightened clinicians, we've got some fantastic people working with us who are already putting the results of projects which are not concluded yet into work in the clinic. So there will be an impact, but it's not going to be general. But as soon as you produce some evidence, that's when people will start to believe. So the sooner the evidence comes about, the quicker it'll happen.
You've obviously been focusing on some of the areas in medical and disease.
Yes.
I could see other applications of this, for example, in things like sports performance.
Yeah, absolutely. Yes. Yeah.
Are you working on any of those? Or is it
No, we only have 24 hours a day, unfortunately. But no, we're very interested. I mean, that's one of the thoughts we had when we started getting involved in musculoskeletal. We never found a sports partner of a similar mind. We've worked with a number of partners now for 10, 12 years, and you begin to understand how they work, and you get a very, very good rapport, and then it's easy to work collectively. When you bring a new partner in, it is a learning. It's like a marriage, I guess. Get a rocky few years at the start before you settle down to your final blissful happiness. One hopes.
This project is clearly not just national but international.
Absolutely. Yes.
How do you control things like standards and who's doing what to make sure that we're not duplicating work done somewhere else, or there's a big hole in the research which needs to be filled and so forth?
Yeah, very good question.
How are those sorts of things
Very, very, very good
handled?
question. Absolutely critical. The Commission has set up what it calls the VPH Network of Excellence, and that has the responsibility of having an overarching view of what's going on amongst all the VPH projects. Okay, so it's setting up a toolbox, for example, into which everybody contributes. That will be open source, so available to everybody, and the people who are creating this are trying to ensure that the intercommunicability of the various components of this. What you find is that some partners are involved in more than one project, and therefore naturally they are introducing standards which may be informal rather than formalized standards, but will span several activities. We're also in great dialogue with the Americans. For the first time, the Commission has actually funded transatlantic collaboration with European money only to ensure that what's happening in the NIH in America in comparable projects feeds into what's going on in Europe, to ensure that we're not doing things that are out of step. So it's not getting to the stage where you're going to standards boards and getting formal written standards, but underneath it all there is a strong realization that this has got to happen, and people are taking a grassroots approach, if you like, to ensuring that it does.
Another very quick point. You said it's all going to be open source. Are you talking to people like the Open Software Foundation to make sure it's more widely known that it's open source and available?
Personally, I'm not, but I would almost certainly guess that the toolkit people are, because the toolkit has been organised at Oxford. I don't know, they've been doing it for a number of years now, so I would imagine that they definitely are.